Good morning, my dear students. So today we are going to continue with the second part of the insect pest of legumes. So in the last class, we were discussing the important pests that are causing economical damages on the legume crops. So because legumes are very important for all people living around the world because they are the not only the protein sources but also the so many vitamins and the other medicinal values are there. So therefore the studying of the pest on targeting on the legume crops are very important. One of such pests is spotted pot borer. Spotted pot borer, we call it as an Maruca vitrata. This is a new name. So earlier it was called it as Maruca testulalis and Cruciphora testulalis. There are different names, so it is distributed all around the world. So this because there's a power cut, but I can continue. But so there is this distributed all around the globe, but this is not a major pest. But some part it is mentioned in the red spot on India and some of these. So the Chinese part and the Korean part. So this is called it as a major pest. But nowadays, because of this mismanagement, this is also become as the major pest of string bean. So sometimes we call it as a sting bean caterpillar. So because it's damaging so much of pods when the pods become mature in the string bean. So that's what we call it as. Sometimes we call it as a sting bean pod borer. So you can easily identify because this is a very active adult. So when you are rearing the larva, when the larval rearing, you will learn how these behave because it's like a jet flights. It will fly very fast. So adult moths, right? So this is an identification of the adult moths is wingspan of 20 to 25 millimeter. So 20 to 25 millimeter means so you can find it. So how much of length, and also this is easily you can identify based on the only two color mixes, the brown and the white, right? So brown and the white. So in the head is a brown color, abdomen is a brown color, and the eggs are large, right? There are larger eggs, so you can easily identify. Easily you can see when you are observing the adults very closely. So the antenna length, right? So antenna length is the length of the forewings. You can see the antenna touch the tip of the. So this is we call it as the apical margin of this wing, right? So it will touch where these wingspans are there. The forewings are elongate and conspicuous trans, right? So this is the identification character, right? A specific identification character of the adult. So because you can see this is the inverted. So because sometimes we call it as a transverse conspicuous white along marking, right? This type of marking would have seen in the uh, Cine coat semi-looper 2. This is we called as a silvery marking in over there, but here in white, right? Here in the white. So eggs, the females are laying eggs small eggs individually, mainly individual. Sometime in some cases you can find the small batches on the flowers and the flower buds, right? The flower and flower buds. So they lay on the terminal shoots of young plants too, right? So incubation period is 3 days to 24 to 27 degrees Celsius. So some it is varies with temperature to temperature. So egg are ovate, pale yellowish, white, and translucent. Translucent mean is like a uh, sagopal. It's like a sagopal, but not a pure white. It's a translucent, right? It's a translucent off-white color. A slight reticulate sculpturing on the Korean. So sometimes you can find Korean also if you're observing the egg under microscope. Size of the egg varies with the fertility fertility of the capacity, fertilizing capacity of the female. So sometimes it's look like a droplet of water because maybe it's a transparent. That's what I told you. So after the cooking of this imiocarp, this sago pearl, how it will be the individual sago pearl after the boiling, the egg would be like that, right? It's an easy identification. So lava. So now if the females are laying eggs, if the females are laying eggs and flower buds and the flower terminal junctures, the initially after the catching, is early, the neonatal, so they are making the webs, right? They are making the webs. I can show you, right? They are making the webs here, like these webs, and eating inside and damaging these flowers and the flower buds. So that is a problem. And also sometimes the larval character is it makes a webs in by using 2-3 leaves in the shoots and make the leaves to become as a web, right? Like sometimes you may confuse with the spider webs. So inside that you can find this creamy initial lava, right? So the early stages of the early insta lava is a greenish transparent like the color, but it's very active. So you can easily identify the identification of the character is you can see this brownish prothoracic shield, right? So prothoracic shield. So this lava is also very active. If you are touching from the head, so it will move backward. If you are touching in the backside, it will move forward, right? So both way it can move, and it is very fast moving, right? It's fast moving lava. So once once the lava growing, so it will reach finally the full grown lava is nearly about 17 to 20 millimeter long. The fully mature lava will become a sometimes dark greenish to brown, dark greenish to brown. So this is head capsule is light dark brown and prothoracic plate is a dark brown. So body white is to pale green. That's what pale brown. That's what I told you, right? It's mature lava is become like is greenish to brown with irregular black spots. You can see irregular black spots in the dorsal, the whole dorsal body you can find. The spot become indistinct immediately before pupation. So pupation means lava become, you see, so in this stage, after this stage, this lava is going to become a pupa. A lava is going to become a pupa, so therefore it become a darkish brown color, right? It is barking from the 5 larval insta will be there. 5 larval insta will be there. So lava, so how it is attacking the first insta lava may be found together among the flowers. So thereafter disperse singly and moving one flower to another flower. Each larva damages 4 to 6 flowers. You see, if a one larva damages 4 to 6 leaves, 4 to 6 flowers, if 20-25 larva is there, then you can imagine that it can finish the whole plant. It can finish the whole flowers. Your plant will give any yield. So young larva may feed a part of the flowers and the foliage. If the flower is damaged, right? So you cannot expect it to produce the pods. But later insta larva it's more common in the pots, right? So once the larva grown, fully grown larva is make a round cut and go inside the pots and it will grab the seeds and it will eat the seeds, entire seeds, right? Older larvae bed together flowers and the young buds and feed pupate inside the webs. Once it's fully grown, it's going to pupate. What is the behavior of this larva is making the web by young pods and the flowers, and inside this web, it will pupate, right? On quite hard pods, large feed exclusively on flowers. If they are a hard pods, so larva feed on the flowers. Larval insta is 13 to 14 days under optimum environmental temperature. So this is the pupa, right? So this is a pupa. This is an initial pupa. Is greenish to brownish color. But in the latest stages, it become a very brown, right? Very brownish color. It's an optac type pupa. So you can see that 1.5 into 2.5 millimeter silken cocoon. Inside that, you can see this adult, right? So how you will manage this one is a chemical application. Farmers nowadays, they are using the chemical applications at the time of flowering. So if, because they are females laying eggs and once the flower bud initiation, right? If the flower buds initiation start at the females started to lay eggs. So at the time farmers, so if they are applying that, so commonly farmers, they are using the netabiron. So this is a chemical, but they can use for the molting interruption chemicals, novel chemicals, fifth generation chemicals also. Some farmers, they can use chlorofluoxuran and novelirol. So these are the chemicals, they are molting disruption chemicals. So I mean, once the first instar larvas, I mean, there is no mold to become another stages. If it is a pupa, no adult. So this molting interruption chemicals, so you can, the farmers are using. So time of plantings. Normally you can Maruca, you can expect the December to January, because the farmers they are cultivating this sting bean in the Maha season, fully Maha season in many part of the country, because in that full high rainy season, so they will not cultivate any other vegetables. So at this time, so this Maruca will be very active. Therefore, so they can shift the calendar. They can shift the calendar. Lambidus and Calicovopa. Calicovopa is a pot borer we studied in the last class. And Lambidus mean you will study from the next slide. So Lambidus mean blue butterflies. Lambidus barticus. So Catocrypsis. So these two are active in February and March. So if the farmer, you see December to January, February to March. So in these 4 months, there will be a problem for cultivating these string beans because of this one to another, one to another. So these pests will be active. So therefore, the farmers can shift the calendar before set of rain. So before December, if they are cultivating, or after the March, so they can escape from this pest attack. So resistant varieties, there is no any resistant varieties, but the conservation of the natural enemies, limited application of the chemical or sorghum, cowpea, intercropping many times the borer population. So why? So these monocotton dicot. Why? So monocotton dicot plant. So you are cultivating that alternatively or intercropping. It will confuse the pest. So there will be chances for less attack of the pest. Biological control also there. So this for Maruca tessularis and ATLA singanilla. ATLA singanilla is another pot borer. Also green lentil pot borer you will study in the forthcoming slides. For these two, the parasitoids are there. So Ancylostomia strecoria, then Bracon cajani, and Emphysema dentata. So these three parasitoids has been reported, it is there, but the problem is if you are going to augment these parasitoids, then you have to omit or limit the chemical application. Otherwise, it is will not survive. So because the biocontrol agent are very delicate, so you should not apply. But if the environmental temperature is not suitable for the biocontrol agent, so it won't work. There is a problem, but it is introduced, these three introduced in 1950s from Trinidad, which is West Indies, Trinidad and Tobago, and Mauritius to Sri Lanka. Partially successful, 40 to 70% increase the harvestable yield. But introduced in 1950s in Hawaii, 1970s in Sri Lanka, and 1970 to 67 to 70 in Fiji Islands. So these three parasitoids introduced many countries, so and partially control this spotted borer. But nowadays, these spotted borer are very big problem in throughout the string bean cultivation. So because of this unavailability of these beneficial organisms, because we have abolished or we have killed these beneficial parasitoids by over application of these broad spectrum chemicals. Another one is the, so this is, I told you, the lampidos. So these are very active to March, right? February to March. Catocryptus, there are 3 species in Sri Lanka, right? Catocryptus nigus, or Lambidus boticus, or Catocryptus strabo. So these 3 species are reported in Sri Lanka, but these Lambidus boticus are common, right? Lambidus boticus are common or highly available, or frequently you can see when you are examine this blue butterfly. This is an identification character of this blue butterfly. So butterflies, you can see there are 2 conspicuous spots on the hind wing, right? Anal margin of the hind wing. You can find the 2 conspicuous spot. So this is a blue color butterfly. That's what we call it as a blue butterfly. And also you can see the hind, in between these 2 conspicuous spot, you can find the loop-like structure, right? Loop-like structure. Then another identification character of the blue butterfly. So Lacanidae lepidoptera. The adults are not pests. Yes, you all know the lepidoptera, except the fruit-sucking moth, other adults will not cause any damages. But here also larva is a problematic. But compared to the Meroca testulacis, this lamboides attack, the blue butterflies attack is less. So you can see, easily identify, you see the capitate antenna is an identification character. This is a butterfly. So this is a loop, anal hind wing loop. And this is the larva. You see, in between the flower buds, larva will be there. It is very, it's not differentiable. You see, it's look like a flower bud. This is a larva, but it is very sluggish larva. Movement is very, very slow. So you can easily touch or easily trap this larva or easily catch the larva, right? So this larva, you cannot differentiate sometimes whether it is larva or flower buds or seeds, right? So larva, because that's the thing. Sometimes you can find this larva may become a greenish color too. There are 2 colors. Sometimes this is a creamy color. Another one is the greenish color. Right, this is the larva. You can see there is no any conspicuous marking. Only you can find a short hairs, right? On short hairs. This is a pupa. So pupa, you can find it is the fallen leaves with the pupa or pupate inside the fallen leaves, right? So you can see these are chrysalis type pupas because of the butterfly. You can see easily differentiable the chrysalis type pupa. So adult lay eggs on the flower and the flower buds. After catching, tiny caterpillars enter into the unopened flowers. So they are flower feeders. If they are a flower feeders, mean this is dangerous. Why? Because before pot initiation, if this number of larvas are high per flower for plant, so they can finish off all these flowers. If it is finished off, then it's highly economic because these 2 are damaging economically important part of the crops. Therefore, you have to be very careful. So from the flower bud initiation, you have to monitor closely to manage these 2 pests. Pupate. Sometimes you can find it's a pupate within spots, but normally you cannot expect. So because it's fallen leaves and in the very close, the pupate in the underside, the bottom part of the plant closer to the soil. So this is the last caterpillar. Right? So this is another caterpillar. It's a lentil pot borer, Atilla singanilla. This attack is less in Sri Lanka. Right? This attack in Sri Lanka, but this Atilla singanilla has been reported in India, southern India, and throughout the India and the Bangladesh and other countries. This attack is very less. So adult lays on the lay eggs on the flowers. Emerging larvae feed flower. This is more or less similar to the feeding behavior and the feeding pattern of the Meroca testulacis. Only the pupal color and the adult, the larval color and the adults are differentiated. Pupate within the pods. So this is the life cycle of the lentil pot borer. Atilla singalna. You see the adult color, right? Is a brownish color. Initially, the larva reddish color, it will become a brownish, but you cannot expect any conspicuous brown marking, but you see there are 3 spots in the prothoracic. So prothoracic shield is brownish color. From this you can easily identify. Right? This is an Atilla singanilla pupa. And another minor pest of these are the minor pest of legumes. One is first one is bean aphids. We call it as an Aphids crucivora. This is a polyphagous one. So where you can expect, because we are going to cultivate the string beans, so mainly the legume crops in the Maha season, the before just onset of rains, you can expect these aphid attacks, right? So they are nowadays, these aphids are becoming the major pest because we have wiped out the beneficial parasitoids by the over application of pesticides. So therefore, these naturally available parasitoid food that are laying eggs on the aphids are very less. That's how nowadays aphids and mealybugs are very big problem. So therefore, if you are augmenting if you are giving an opportunity to augment these beneficial parasitoids, so these bean aphids and mealybugs will become a minor pests. So right here in the aphids, so you know this is hemipteran, so there are nymph and adulta sucking the seps, right? From the tender leaves and shoot, resulting in twisting of leaves and poor pot development or devitalization of the plant and scooting. This is a problem of aphids. So because if they are an abundant, so lot, so they can cover whole the aerial part of the plant, continuously suck the sap from the leaves, branches, and soft part of the plant, and continuously sucking the plant, so the plants will become withered away. So crinkled leaf and flowers will become a very tiny and crinkled. It will not make any pots. So that is the problem. So if you're observing that when initially it will colonize as a group, so if you are observing continuously or every day in the farming, you can scout, you can remove that one, you can burn it, right? You can burn it, but natural parasitoids are there. So polyphagous, so in any other crops, so it can act as also can use spraying the tobacco decoction. So tobacco boiled in 10 liter of water, half of it and the makeup with 30 liter plus 100 gram of soap solution. So we call it as a tobacco decoction. So this is consisting of neonicotinoids. So you can, if you are applying that one, you can remove that, you can kill partially the aphids, but systemic insecticides are effective if it is an abundance. Through the, with the, earlier it was there as a dimethyl, it's a very good chemical, now it has been banned because of this residual nature. Nowadays other systemic insecticides are there. So this systemic insecticide can work, but it is not recommended to apply aphids because so aphids are minor pest, you can manage easily. You don't want uh, to convert this minor pest to be a major pest by over application of these chemicals. So another one is their green gram potbug. This is also the minor pest. The problem is they are sucking the sap from the pots. So if they are sucking the sap from the pots, so the quality of the pot will become less. Therefore, you have to be very careful. But they are minor pests. There are 3 different types of bugs. Clavigrella gibosa, Reptotus linearis, Anoplochamus pesiana. There are 3 categories of these pot bugs. The problem in legumes. So they are Corridae hemipterans, right? The hardened nymphs, you can see this Reptotus linearis. So Clavigrella gibosa, Anoplochamus pesiana. So Anoplochamus pesiana. So these 3 are right. The three trees are the pots. So their target is to suck the sap from the pots. Therefore, you have to be very careful. So their shoots and fade pots reveal seeds with a dark patch and lost germination capacity. If they are an abundant in the field, then you can expect some of the yield reductions. But so everyday monitoring and chasing out these bugs, you can easily minimize the incidences. Sometimes carbaryl is recommended, so 10 kilogram per acre. So carbaryls you can expect. So this is a broad spectrum insecticide. So you can use it. It's a carbaryl. One you have, if you are applying this one, many other insect pests also you can easily control, but it is not recommended. So because this is also the minor pests, you can easily manage them by using the catching and killing. You can use an aerial net, trap that one and kill it. So another one is the red gram cowbuck, right? So Oxyrachis tendus, we call it as an Oxyrachis tendus membracidae, right? Membracidae hemiptera. This is also the minor pest, very minor pest compared to the other aphids and potbug. This is also there somewhere else. So nymph and adults are sucking the sap, and so this is excrete honeydew. Therefore, it can attract the ants and scutimate development will be there. So if in the aphids, this cowbug and mealybug attack is there, definitely you can expect more amount of ant because they are producing honeydews. These honeydews leads for the development of the scutimate because mainly we are cultivating in the December to February. So this is a humid condition, so there are more chances for development of the scooty. Therefore, you have to be very careful on these days. So you can use sometimes entomopathogenic fungi and bacteria to manage this because in this humid condition, this entomopathogen can work to manage compared to the application of these chemicals. But only the number of insects are high, the chemicals are only weak. Right? Um, so this is one of the major pests of red gram. Red gram is not here in Sri Lanka much, but in India is a very big problem. But this species is common all over the Sri Lanka, not much problematic. Other one is there, other than the insect pest, you can find then red gram mite. This is a problem, this red gram mite in the hot season, in February, March season. So you can expect. So as we call it as an Assyria kajani, right? Eriophidae acarina, Syria kajani. This is why. So because these, if the environmental temperature is high, so these might populate lot and lot. So they will make a web or over the leaves because it's invisible webs. You are going there close, very close, monitoring only you can see. Scrap the green part of the top layer, over the leaf surfaces and eaten away. Then your leaf will become a whitish color, and finally this is dry and withered away. So there are chances, if the mite population is very high, there are chances for dying of plants. But if you are intermittently apply the sprinkler water, the sprinkler irrigation, sometimes overhead irrigation, it can wash away these mites, or the rains can wash away the mites. The problem is these mites and aphids can transmit the viruses, right? Transmit Pongomia sterility mosaic virus. If it is Pongomia sterility mosaic virus is transmitted, you cannot expect any yield from your plants, right? So this is a very big problem. A single area of it mite can is sufficient to transmit the dose. Single mite is sufficient to transmit the disease. If it is transmitted, then your whole field will become a problem. So disease can be identified by the distant patches of bushy pale green plants without flowers and pods. That is a viral attack symptoms, right? Viral attack symptoms. So leaves are similar, show light to dark green mosaic pattern. So if it is mosaic pattern observed, and you have to remove it, you have to burn it, you have to rotate with it. Other crops, otherwise you will not get the yield. Right? Destruction of the perennial and volunteer, the pigeon pea, right? So sorry, the pigeon pea sterility mosaic virus is there. The pigeon pea and other ratoon growth of the pigeon pea. So you have to remove it. So these pigeon peas act as a harbor for this transmission of these weeds to the other legumes. Growing tolerant varieties and fairly spray of the vegetable sulfur. Sulfur can work to manage these aphids. No need to apply for other chemicals. You can apply dusting the sulfur, so you can easily manage this mite. This is the Azaria kajana. You see, leaves become as a whitish color. You see, this is an adult. This is an egg. You see, here and there are a lot of eggs. This is under the microscope view. You cannot see visually. You need hand lens sometimes to observe the adult. So adults will be there under the webs, right? Adult will be there, so running under the webs. So very active in the day. So and it will become go in the downside and hide away inside the webs. So another potbug groups are there. We call it as a green stingbuck and brown stingbuck, Nisera viridula, right? So Nisera viridula is there. This is also attacking. They are sucking the sap from the stems and pods, right? So adults and the nymphs are problematic. So this attack also less, but you can easily manage this space if you are observing the egg masses here. So egg will be laid as arranged in a systematic manner in the leaves like a hand granite. If you are observing that, it look like a hand granite. Small, 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 arranged it. So if you are observing the egg masses, pluck out these leaves and you are burning that leaves, you are removing the populations. But naturally, while parasitoids are there, egg parasitoids. These egg parasitoids, they can lay the eggs and they can damage the eggs. So therefore, you need not to worry about if you are augmenting these natural egg parasitoids. So this is a green beak attacks. This is not an attack. So how they are sucking the sap from the plants. So these are the things we have to discuss more on the legume crops. So this is in end of this section of the uh, legumes crops. If you won't ask any questions, and you can ask questions. So this is the end of the part 2 of the legume. So the legume crops is now over. So if you won't ask any questions, and you can ask questions.